Welcome everyone, and appreciate everyone taking time from what's obviously a very, very busy week. So I'm sorry this is encroaching a little bit on your holiday period, but it's wonderful to get together. So what we'll do for the next hour is we'll go through a lot of highlights. There's been some wonderful work across ENIGMA in the last year. And really the goal is just to familiarize everyone with the different things that have been going on. So we'll go through a number of topics. We'll give you a quick update on working groups and grants, methods and tools and publications. There'll be some separate updates on DTI and functional MRI, and also some other analysis methods and harmonization tools that are being used across the different ENIGMA working groups. And there'll be a little bit of an update on machine learning and also meetings and conferences. And we'll end with some announcements and reminders of materials that might be useful for different working groups. So just a quick summary, all of you as working group chairs are very familiar with this, but just to recap maybe for some new folks, here's a map of the working groups in ENIGMA. There's about 50, maybe 51. They're organized, as you know, into working groups focused on psychiatry, neurology, neurodevelopment, even infectious disease, and also algorithmic and methods development working groups, the list up there at the top, that focus on analyzing and developing methods to process those different types of data. There's a new group on neuromodulation led by Taylor Kuhn. I'll tell you a little bit about that. And so anyone interested in integrating neuroimaging and neurosite data with neuromodulation, things like deep brain stimulation or transcranial magnetic stimulation and so forth, it's very welcome to contact Taylor. They've been having a few kickoff calls to talk about how you would study these different modalities of intervention and their effects on the brain. There's a new group on Tourette syndrome led by Perry Pascu. In fact, they've had 2 or 3 teleconferences. Their R01 grant with the NIMH has just been recommended for funding, so congrats to the team. And this obviously has connections with many other neurodevelopmental disorder groups, OCD, and conditions such as autism spectrum disorder. And so there's a lot of synergies, obviously, between their work and many others. Congrats to Odile van den Heuvel and Chris Friend for their award of a grant to look at task-based fMRI in OCD. That's a new grant that's been awarded from the IOCDF, and looking forward to the activities on that one. The pending grants, these are, we hope, will be funded soon. There's one by Raj Mori and Leon Schmal that scored 2nd percentile. Congrats to them, to look at resting state fMRI in PTSD and MDD. A number of other groups on bipolar disorder, 22q and CNVs are up for funding at the NIMH January Council. There are a number of R01s that have been submitted on pediatric brain trauma, environmental effects on the brain, anxiety, PD, and also an effort in Pakistan to look at brain trauma by Maheen Adamson that are all on their way to being revised or resubmitted. Here's all the active grants. I just thought it'd be... NOA. Oh, I thought it'd be interesting to just review. Oh, there's a little bit of an echo. I thought it'd be interesting just to review all of the funded grants. There are 20. Thanks so much to all of you that have contributed to these. 6 are newly funded in the last year. So there's a Brain Initiative grant to Peter Kochanov to look at schizophrenia, bipolar, and MDD with a measure that he terms the Regional Vulnerability Index. Odile's grant on task-based fMRI we just mentioned. Jana Marie Bass Hogendam and Danny Pine have won an award for ENIGMA Anxiety, a Rubicon Award. Rebecca Santelli's won an R01 to support their Origins group work on infant development. And then 2 R01s from NINDS for the Parkinson's and epilepsy working groups, and congrats to the folks who've recently been funded. A quick summary of methods and tools. So I just thought it'd be useful to go through some of the tools that the ENIGMA groups are using to look at structural MRI, diffusion MRI, resting state, and even MEG and EEG. Many of these methods are being used very widely, so I wanted to review them. Odile presented a slide the other day on a kickoff call for the Tourette syndrome working group, just to describe the way that their working group has progressed in its use of different analytic methods. And so they have a very nice review paper that's in human brain mapping, talking about in the last 5 years their use of morphometric measures using FreeSurfer, some DTI, some cross disorder studies that they've been leading. But then it's moved more recently into approaches for whole brain morphometry, VBM, resting state, and some scripts that I'll talk about in a minute for allowing subnuclear segmentation of the thalamus, the hippocampus, and amygdala, and cerebellum, and now task-based fMRI. I do want to say that Odil's initial consortium, OBIC, did use BBM, so it's not entirely new, but this has represented a progression that many of the groups are doing. So just ongoing projects. The last year saw 59 ENIGMA journal manuscripts. How do we know? Well, you can search PubMed and remove the 29 or so that were not to do with ENIGMA. And this is not counting conference papers or abstracts. It's been a very productive year. Most notable is a special issue on ENIGMA. So human brain mapping published 32 papers. This officially went live in the last week. It's the January issue 2022. And I just want to thank all of you and the guest editors and Gary Egan and Peter Kochanov for the original idea to do this. It's a really great possession forever, as Thucydides would say. It's a possession forever for all time. It's a wonderful booklet that goes through all of the achievements of the different working groups. I want to mention Emily Dennis, David Tate, and Lisa Wile's special issue in April. Their brain injury working group is essentially as an umbrella in which groups focusing on pediatric and adult brain trauma, intimate partner violence, spectroscopy, and also military and sports brain trauma. They all come together and they meet very regularly, and they've, in this special issue, reviewed some of the challenges and successes of their recent work. So congrats to their group for this special issue also. There's many additional reviews, even outside of those special issues. Masoud Tamasian, Kai Spiegelhalter, and Moshtaba Zareh reviewed ENIGMA Sleep, very, very nice review there. Also, Jess Turner, Vince Calhoun, and Theo Van Up have a nice review on the use of CoinStack, a federated infrastructure for computation on distributed brain data that they're piloting in schizophrenia and a number of the other working groups. And then also just a shout out to Yoonjin Chun, who with Theo Van Up recently reviewed the work of schizophrenia, bipolar, and major depression in a nice review paper. Some highlights. Many of you are familiar with the ENIGMA t-shirt that has a number of disease-specific maps on. In the last year, we've seen developments in Parkinson's disease, PTSD, anorexia and eating disorders, clinical high risk for psychosis and schizotypy, and also rare genetic conditions. And I'll just give you a little bit of a tour of some of those recent findings. So one of the most recently funded working groups on Parkinson's disease is led by Isbrand van der Werf, and this paper by Max Landsma and Joanna Bright and a huge number of colleagues across the world found stage-specific thinning and changes in cortical surface area across the 5 stages, the Hoehn and Yahr stages of Parkinson's disease. So congrats to them on this achievement. There's also been progress on the ataxia front. Ian Harding and his colleagues published in Annals of Neurology a superb paper mapping the cerebellum. So as you can see here, the degree of deficits in Friedreich ataxia depend not just on the duration of illness and the age of onset, but a number of other factors where they've done regional cerebellum mapping. And the tools they developed have been also used by many other working groups. So Ian and his colleagues developed a voxel-based morphometric pipeline for studying the cerebellum. It will also parcellate it. It's based on existing and widely accepted tools such as SUIT and other methods. OCD, epilepsy, TBI, Parkinson's, and even 22q are looking at the cerebellum using this software. And there's even a genome-wide association study that's being led by Sarah Medland and Ian Harding to look at these. So do check it out. Also, Tiago Rezende has written a beautiful pipeline for looking at spinal cord segmentation. Very, very interesting for use in different working groups. I also want to mention that Gemma Modinos and Andre Ellerman and Matthias Kirchner published on schizotypy. So this is the first paper from their working group. Despite the subtlety of the effects, you can see this effect has a Cohen's d of 0.07. It's one of the most subtle effects of any working group. It really just shows the power of clubbing together to identify the robustness of these findings. So congrats to them. Also, a predictive model was developed by Maria Jalbozikowski and Dennis Hernaus and their colleagues in the Clinical High Risk for Psychosis Working Group. They combined data from 31 cohorts worldwide and were able to confirm that the regions of interest in the superior temporal cortex were predictive of whether or not individuals would go on to develop psychosis. So very, very important work there. The Suicidal Thoughts and Behavior Working Group, led by Lian Schmal, Neda Jahanshad, and their colleagues, published on brain correlates of suicide attempt across 18 international cohorts. Very, very important work. There's been efforts also on understanding clinical and neuropsych measures of suicide-related behavior, and a nice paper that's been published on those. I do want to mention that also efforts to harmonize neurocognitive and behavioral data emerging in many of the working groups. Eamon Kennedy and the Brain Injury Working Group submitted a manuscript on clinical scale harmonization in brain trauma to a special issue. Now, I talked with Emily Dennis, who's one of the editors of this yesterday, and she says if you have any papers on harmonization, whether it be imaging, genetics, or neuropsych or clinical data, do consider sending them to that special issue. It's about to close in about 3 or 4 weeks, but the door's still open for that. Very nice new paper by Esther Walter and Stefan Ehrlich and colleagues studied individuals with eating disorders and anorexia nervosa. And this is among the largest effects on the brain that we've seen in the psychiatric working groups of ENIGMA. They just submitted their first report on it, and congrats to the team for that very important study. Masoud Tomarsian and Kai Spiegelhalter and Mojtaba Azare have been spearheading an initiative on sleep, where they're beginning to relate actigraphy and polysomnographic data to imaging data. And if you have interests or expertise in that arena, please do contact them. Sukley Liu and their colleagues have been publishing initial papers on outcomes in stroke, finding morphometric correlates that are predictive of outcomes in a very large data set. And they have a number of projects, including crowdsourced lesion identification to train machine learning algorithms, and a number of different activities. So congrats to Lee, Artemis, and those involved there. Lauren Salmonen, Megan Herting, and JC Chen it developed this really terrific working group looking at environmental effects on brain health across the lifespan. They've assembled 45 international cohorts that either have or are willing to geocode their data, which enables the brain scans to be rated to pollution and environmental health data. So if you have an interest in that area, please do contact the PIs. Also, Stefan De Brito and colleagues listed here, Moji Agachani, many others, Nidian Gao and Marlena Staginos, are drafting a review on their working group for translational psychiatry and just completing their first morphometric study in 21 cohorts across Europe, the US, Brazil, India, and China on antisocial behavior and conduct disorder in youths. And so congrats to them on their first publication just being finished. Very important initiative led by Sven Muller and Eileen Luders is looking at sex and gender differences in the brain. So there's an excellent review written by Lawrence Salmonen on the differences in women and men in the incidence and progression and etiology of the different disorders we study. But this is a very interesting paper on gender, looking at cis and trans women and men and the different neuroimaging findings that are apparent in those different groups. Also, ENIGMA Laterality, this has been a bumper crop of studies from Clyde Frank, Shangzhen Kong, Simon Fisher, and more recently, Eileen Luders has generously joined as a co-chair for the Laterality group. They've been studying evidence for abnormal lateralization in a number of working groups based on the curated and QC'd data from ASD, MDD, pediatric and adult, OCD. And I just want to thank them for their tremendous work looking at all of the subtle perturbations of laterality that they've been publishing in these different disorders. One of them that I think is very notable and interesting, all of you working in psychiatry are familiar with the story in the early 70s where Eve Johnston, who's a very prominent psychiatrist in Edinburgh, was the first to identify ventricular changes in schizophrenia using CT in 1976. And soon afterwards, there was a hypothesis by Tim Crow that in schizophrenia, lateralization was altered, was diminished. And this wonderful new study by Dick Shiven and colleagues working with Clyde Franks in the Laterality group and Jess Turner and Theo Van Oort in the schizophrenia group has essentially found evidence to support this. It's very subtle, but this notion that there are, in particular in the middle temporal gyrus and some other regions of the cortex, areas in which people with schizophrenia differ in laterality has confirmed a 30-year-old hypothesis. So these studies take a long time, but that was very, very, very exciting to see that work coming out. DTI, I want to extend a note of thanks to Neda Jahanshad and Peter Kochanov, who've overseen a large number of diffusion imaging studies from ENIGMA. They're all tabulated here. So even in the last year, there's been studies of PTSD, OCD, addiction, bipolar. The different subgroups of TBI have published, along with Parkinson's, autism spectrum disorder, and early onset psychosis. And then some newer projects looking at rare genetic variants and DTI. I'll just give you a quick update. So the OCD group led by Odile van den Heuvel and Dan Stein has a sub-project, a consortium known as Domain, looking at DTI and OCD, in which Fabrizio and Federica Piras and Gianfranco Spalletta meta-analyzed data from numerous sites and found a number of very interesting things. The FA, the microstructural measure FA, was altered, but this also shows the power of multisite analysis. They found that the degree of difference on DTI depended on the duration of illness, the age of onset, and even on the fraction of individuals medicated in the different cohorts. So this gives us a lot of interesting things to follow up in the future. Now we're identifying these modulators. Claudia Barth and Sinead Kelly completed a DTI analysis of early onset psychosis. Now this is relatively rare, only about 0.5% of individuals have early onset psychosis. But they were able to find really quite strong effects on white matter microstructure, with largest effects in the superior longitudinal fasciculus, Cohen's D around 0.37, really getting into the differences and whether they overlap with idiopathic schizophrenia also. A brand new hot off the press project looking at DTI and Parkinson's, this group again led by Ismail van der Werf, but Conor Owens-Walton and many groups around the world, in fact 16 international cohorts have shared their diffusion data to find that as the disease progresses, there's a progressive cascade of events in which greater numbers of regions of interest and with greater intensity, you'll find effects as Parkinson's disease proceeds. The addiction working group led by Scott Mackey, Hugh Garavan, and Patricia Conrad published a really interesting study on different types of substance use. So they looked at white matter microstructure in people dependent on cocaine, methamphetamine, and nicotine. Excuse me, and they were able to confirm and extend what was known in terms of microstructural differences across the brain, finding both unique and distinct patterns of difference in people who are dependent on these different substances. Also 3 more DTI papers, one by Emily Dennis and her colleagues in the pediatric TBI group, found that acutely post-brain trauma, there were really quite severe differences in microstructure that somewhat encouragingly moderated, at least in a subset of patients later on. And I think understanding these factors and mapping them out with respect to duration since injury is a fascinating topic. Heather Bouchard, working with Raj Mori, has been extending the diffusion analysis to multivariate methods. Many of you are familiar with non-negative matrix factorization, which is a method to look at subtypes in multivariate data to identify biotypes of PTSD that may have different clinical characteristics. And then finally, moving into multi-shell diffusion, Julio Villalon, working with many others in the CNV and 22q working groups, have been doing dimension reduction PCA on multi-shell diffusion metrics, finding patterns or latent groups of features on diffusion that are different in carriers of different copy number variants that put individuals at risk for developmental and psychiatric conditions. So moving on, there's a little bit of an update on functional MRI. So many of you are familiar with ENIGMA Half-Pipe, which was developed by Leah Waller, Ilya Veer, Suzanne Eric, and Henrik Walter. This is a tremendous toolkit that's been piloted in a number of the working groups for analyzing both resting state and task-based functional MRI data. I want to thank the Berlin Charité group and Ilya, who's now back in the Netherlands, for supporting this work with a number of tutorials. And also just to look at the progress of this, the first analysis was finished by Carissa Weiss and the PTSD working group, looking at both static and dynamic functional connectivity with resting state MRI. And these tools are now being piloted both in OCD and MDD, also schizophrenia. Ali Maleh and Theo van Erp just submitted an abstract using this in schizophrenia across 6 sites. And also as part of ENIGMA Brain Injuries R61 grant, this is being piloted by Frank Hillary and Emily Dennis as an approach to look at functional activation in people recovering from brain trauma. This work, very, very nice work in Carissa Weiss's thesis, actually has a web app that'll take you through different measures that she found altered. These are graph theoretic measures of functional connectivity in people with PTSD. And there's both static and dynamic differences that you can look through if you look at the web app that she developed. So a little bit of an update from Leah Waller, Ilya Veer, and the team. Halfpipe is open source. Please do have a look. There's a live tutorial on how to use it. There's manuals for both task-based and resting state fMRI and also QC, and a preprint that you're very welcome to look into. A quick update on other image analysis methods being used across ENIGMA. There's a lot of interest in surface-based and vertex-based mapping of morphometry. There's a study up on the top left there by Lei Wang and Boris Gutman, mapping the regional morphometric abnormalities in the subcortical structures in schizophrenia. And then also a new one just submitted by Max Lansma and Isbjørn van der Werf, identifying a number of morphometric abnormalities, very subtle, in the basal ganglia in Parkinson's disease. Now, this is a truly remarkable study. So Kuldeep Kumar and Seb Jacquemont and the CNV working group, Chris Ching and Kerry Bearden, have been charting the effects of rare genetic variants on subcortical morphometry. You'll see the profile is very different depending on the deletion or duplication that you have in the genome. And they're very interested in whether the profile of morphometric abnormalities is similar in the disorders which these genetic variants put an individual at risk for. So they're very interested in reaching out to groups that have completed a shape morphometry project to do a cross-disorder project. And just to be very specific, the 11 copy number variants or genetic variants that they've been mapping, it's interesting to see how they relate in their profile of abnormalities to, for example, the differences in major depression. A nice paper by Tiffany Ho and Boris Gutman is just out in Human Brain Mapping, showing mild but subtle abnormalities of the hippocampus in terms of structure shape. And then also in schizophrenia, very much more widespread abnormalities with greater effects that you can see mapped there. Again, that paper led by Boris Gutman and Lei Wang, and it just came out last week. It's the very final paper in the Human Brain Mapping special issue. There's a lot of interest in surface-based machine learning. And so there are a number of efforts that apply deep learning techniques, particularly graph-based convolutional neural networks, to vertex-based data. You'll see the ENIGMA MDD working group has projects led by Martin Poirot and Lisbeth Rienemann, Lingli Zhang and Chris Ching working in bipolar, and Vladimir Belov and Roberto Goya Maldonado are also working on these methods in MDD. And Laura Han is doing a slightly different project predicting outcomes in MDD based on these methods. I just want to say, if anyone has an interest in this, there will be a call in the new year that we'll arrange, or the MDD group will arrange, just to brainstorm in different ways that surface-based data can be used for machine learning and prediction, particularly as it's more convenient to transfer across sites in some cases than raw T1 weighted images. So we're excited about the promise of this. All the cortical studies also have vertex-based data. So I want to give a shout out to Xin Wang of the University of Toledo for his work on vertex-wise mapping of cortical data. He's now working with Raj and with Aris Sotiris and Jin Yang on subtyping. So you can apply non-negative matrix factorization to data from people with PTSD. And the interest here obviously is to find subtypes that may have differences in clinical outcomes and characteristics based on clustering and subtyping the data. There's also a project on cross-cultural MDD. So Lian Schmal and Yara Tondas are working with Xiaogan Yan, who leads a Chinese consortium studying resting state data in MDD. And they're also beginning to look at, including software that Xiaogan himself has developed, methods for surface-based mapping of functional data and structural data in major depression. There's tremendous work that's been done by Matthew Kempton and Tony James, initially in partnership with the OCD working group, but now in early onset psychosis and PTSD, to do voxel-based morphometry. And if you're interested in this, do get in touch with Matthew. There's a little bit of an example here just to show how this works. The projects that have been pioneered in the OCD working group have enabled people to keep their raw T1 data in their lab or wherever they're used to analyzing it, and just transfer the effect size maps either to Matthew or to the protagonist of the working group. And I just want to give a shout out to their working group. And one of the things that is tremendously exciting about this work, and this is work by Matthew's team, is that it enables you to understand how results coalesce and become more robust over time as more and more studies are added. And a huge contribution here by Odile and Dan and the OCD working group in trying to understand how these morphometric abnormalities change and reinforce as more data is added. Okay. Another emerging direction in ENIGMA, which is very exciting indeed, is the use of the ENIGMA Toolbox. And so this is an API or software toolkit developed by Sarah Lariviere and Boris Bernhardt, which allows cross-modal comparisons. In other words, it allows you to relate brain imaging findings to other sources of neuroscience data. So at the microscale, you could look at gene expression or neurotransmitter receptor maps or myelination maps. And at the macro scale, you could look at structural and functional connectivity maps from other projects outside of ENIGMA. So just to sort of refresh people's memory on this, there's a tremendous project that was spearheaded by Yash Patel and Thomas Paus, which it was looking at virtual histology. And so just to say a little bit about what that is, all of the disorders and conditions that had been studied in this paper yielded a profile of morphometric abnormalities in the cortex. And they related the patterns which differ by disorder to aggregated data on gene expression in the Allen Brain Atlas. And because at least some of these genes are somewhat specific to certain cell types, such as pyramidal cells or endothelial cells or microglia, it can give us clues as to types of cellular abnormalities that might be present in the different disorders. Now, this work was built on by Sarah Lariviere and Boris Bernhardt, initially working in the epilepsy working group of ENIGMA, to relate all of these cortical maps, not just to cellular data, but other maps of cytoarchitecture, myeloarchitecture, and multimodal connectomics. And they've published a number of papers, including this one in Nature Methods, that make it convenient to relate the maps that you're producing to any number of other modality data. And I just want to commend Sarah. She won an Open Science Prize for this work that's really been tremendously useful for many groups in ENIGMA. If you want to have a look, the URL is there at the top, the ENIGMA Toolbox, and there's all sorts of different things, including visualization, that this toolbox enables you to do. Also, more recently, work by Justine Hansen and Bratislav Misic have been relating published maps, so often published maps from the different ENIGMA groups to neuroreceptor distribution, other molecular and connectivity maps. And I think this is the beginning of a bridge that can be established between other sources of cellular neuroscientific data and the imaging findings that we're collecting. If you'd like to be involved with these, and so many of these papers selected disorders that were interesting to them, but if you also have data that might be relevant, do reach out either to us or to the authors to have your data included. I just want to say a quick word on harmonization of multi-site data in ENIGMA. There's been a tremendous number of studies over the last year looking at different methods for both statistical and direct harmonization, trying to impute data that maybe some sites didn't collect, or deal with a case where different scales or assessments were used across cohorts. One of the most common techniques that has been applied is combat, and many of you are aware of this, but I'm just refreshing everyone's memory. The UPenn group in particular, and even going back to 2007, has been working with a set of methods that derive measures from images for site effects. So if a scanner or certain site reads a little differently in its mean values and variance, there's a number of methods that can preserve the effects that we care about, but eliminate or adjust for the effects of sight. Just looking a little bit more at this, a really nice paper by Delin Sun working with Raj Mori has tried a number of these methods. So I'd recommend having a look at this paper. It's now on bioRxiv, where they computed mean cortical thickness, as many of us do, from many regions of interest and many sites. And if you look at this here, the raw data being on the left and the data that's been run through Combat and Combat GAM, which is a sort of nonlinear extension of Combat, show a greater degree of harmony. And they also assessed whether or not, and in fact, it was an improvement, whether or not doing this boosted their ability to pick up effects in data from 29 sites. There's also certain pointers to be aware of. If you use just linear combat, it can overcorrect the data, it can eliminate, it can sort of be like Procrustes and make everything linear. And so these papers are worth looking at if you're beginning to do multi-site data analysis, just to see what kinds of things happen if different types of site data correction are implemented. There's a tutorial that you're very welcome to listen to on all of the harmonization methods, at least that we could think of, from combat to combat GAM, COVBAT, distributed combat. I want to commend work by Vladimir Belov and Roberto Maldonado Goya, where they've talked about how you would apply combat in a machine learning setting. Very often you're training and testing machine learning methods on data that you would like to be independent. And they noticed and some important things that you should be aware of if you're doing this to make sure that you don't have data leakage. But there's more information in the video if you want to have a look. Joaquim Radua and his group in Barcelona developed a very nice toolkit that you can see down there at the bottom. You can access the code at the bottom that allows you to apply combat when there's missing data. So many of us know that a requirement for machine learning and many other techniques is that the data is complete. And we do want to be able to harmonize data when, at least some sites might have some modalities missing or some regions of interest missing. So do check that out. Joaquim and his team have applied this with the schizophrenia working group in ENIGMA to data from 33 sites. And this is a really interesting roadmap of all the different ways you can harmonize data from sites ranging from linear mixed effects models through to different types of combat with and without imputation. And it gives you some easy to use tools for doing this if you want to give that a try. Just a quick update on machine learning across ENIGMA. At the last count, there were about 40 projects going on that use machine learning of various kinds. And I'll just give you a few updates on some common themes that are emerging. So one of the machine learning initiatives across ENIGMA is to look at brain age. In fact, there's a dedicated working group in ENIGMA led by James Cole on algorithms for computing brain age. In other words, the apparent age of an individual from their brain scan is estimated using machine learning techniques applied to data from healthy controls. There's a number of ways to do this. One of them was implemented by Laura Hahn and Jan Schmal and their colleagues in ENIGMA MDD. And if you want to use it, you can go to Tim Hahn's website, Photon AI, where you can upload anonymized free surfer data and get brain age measures. And this has been implemented on the right in a number of working groups, along with other in-house methods for looking at brain age. Just to sort of mention two hot off the press studies, Ashley Clausen and the PTSD working group looked at brain age in 21 sites, and they compared not just Laura Hahn's method, which we're calling ENIGMA MDD Photon, but also some other common methods, and found that the brain age acceleration or gap was greater in younger individuals with PTSD. And then more recently, Konstantinos Konstantinides, Danai Dima, and Esther Walton have just completed a study of brain age and schizophrenia. And you can see from the forest plot, really robust evidence that patients have accelerated brain age or greater brain age than matched controls. And they're just about to submit that paper. Also work on subtyping. So this is work by Alana Panzenhagen and Philip Seaman in the MDD working group, where they're very interested in using the derived, in this case, morphometric data to identify clusters. And one of the things I've most enjoyed about listening in on these working group calls are all the methods emerging for unsupervised clustering. This method called Fabia, which clusters the imaging and then lets you go ahead and look if the clinical characteristics of the clusters are different, is one of many that are being pioneered. And I want to thank Philip for his leadership and Alana for her work on this project. Now, many of you will say, well, looking at clusters is a little bit tricky when disorders are either age dependent in their effects or depend on the duration of illness. So you have to be very careful. So in many cases, patients at a different stage of the disease do not simply have a different disease, they just have a more progressed state. So this method by Andre Altman, Seymour Lopez, and Neil Oxtoby is really quite remarkable because it can disentangle, particularly in progressive disorders such as epilepsy and Parkinson's, degrees of progression and subtypes with a sort of spatial and temporal factorization of effects. And so many of you studying disorders that are progressive, or at least age-dependent might be interested in perhaps reaching out to them. It leads to some very interesting disentanglement of processes. Also, I want to commend Shi Zhu, working with Raj Mori and Mark Logue in the PTSD working group, for completing what I think is the first ENIGMA machine learning study that's used data from all modalities. So this used the pipelines that we are all using for extraction of data from T1 morphometry, diffusion microstructure, and resting state. And it went ahead and distilled features using variational autoencoders and used them to see if case control status could be predicted. Now, just looking at how this is done, this is also work with Yuval Neria and Raj Mori again. They found that when you put all modalities of data together and you ask the machine learning algorithm, first of all, just to classify patients versus controls, a number of methods boosted the power. So we talked about combat, which is there on the bottom left. Both combat applied to resting state and MRI and DTI ahead of time boosted performance. This is work that they're also piloting in the OCD working group. But they also found that various dimension reduction techniques from PCA to ICA to variational autoencoders were able to boost the performance for case control differentiation that you see in the graph there at the bottom. So congrats to Shi Zhu and colleagues on this project. I think in a sense it's a roadmap that folks might be able to follow, perhaps consulting with them on how to get this to work. A couple of things to watch out for. So machine learning is both wonderful and a little bit worrying in some contexts. Joaquim Radua has written a paper and also a toolkit to fix a problem that comes up in applying machine learning to multi-site data. So one of the things that many of you will be aware of is incorrect learning of features in a cohort that won't generalize well. And this is sometimes known as the lazy learner problem in machine learning. There's some sort of coincidental aspect of the cohort that machine learning methods lock onto, and then this won't necessarily generalize well to more diverse cohorts. So the multi-site accuracy toolkit is available for any of you to use and try out. This is a more pernicious effect than the one I mentioned about with combat, where it will give you apparent results that aren't corrected properly, whereas failure to harmonize in other contexts can often just eliminate power and give you false negatives. A little bit of an update on meetings and conferences. So there's been not enough in-person meetings. We were lucky enough to have a meeting of ENIGMA Epilepsy in Chicago in December, earlier this month. It was nice to see a number of folks from there. But a big thank you to everyone that's presented virtually. I know this is very difficult and challenging to attend these conferences remotely, but ENIGMA groups and members managed to present 15 abstracts at OHBM, a number of symposia, 4 actually at SOBP, and a number of them at meetings on anxiety, epilepsy, technical meetings on ISB as an image processing meeting, and a number of them are seen here, and thanks to all of you for presenting and promoting results in these different contexts. Upcoming conferences in 2022. Last week, which was why we didn't have this update last week, everyone was a little bit busy, or most people were busy with brain mapping abstracts. There's also been a symposium accepted on machine learning and big data at SOBP that is due to be presented. Peter Kochanov has led a symposium, the Pacific Symposium on Biocomputing, which we hope happens as planned in Hawaii in January. And also there's a number of abstracts in addition to the Ataxia Research Congress that were submitted to OHBM. Apologies if yours isn't listed. I mean, in this short overview, many of you may have done things that we omitted here, and if so, I'm sorry about that. But if you have abstracts for these and other conferences, please do email them to Sophia Tomopoulos, and she'll make sure that they're highlighted and known to others. Do come to ENIGMA, and thank you to David Glann and Theo for suggesting an outfit that I might wear at the Edinburgh conference in Scotland. We hope that's in person. Of course, we don't know. And for many of that have submitted, they're also having a hybrid option. So if it's not feasible for you to attend, or maybe for us to attend, we'll go ahead and do that virtually. A couple of announcements and reminders. So these are things that have come up in recent months that are very exciting. There's an effort led by Emily Dennis, Lisa Wild, and David Tate of the University of Utah to set up ENIGMA U. And it's an online school for neuroimaging and neuroscience, which is aimed at advanced high school and intro-level college students. Now, many of you are familiar in ENIGMA and neuroimaging. You're teaching new students the principles of both neuroimaging and neurological and psychiatric and neurodevelopmental disorders. Emily and her colleagues have actually obtained funding to start to create modules on introductory neuroscience and psychiatry. If any of you are interested in recording a short video, maybe 15, 20-minute video about the disorder you study or any topic of interest, please do reach out to Emily and David and Lisa for this. I think this is a tremendous effort. And it's also possible to match possible students who've done this with ENIGMA hosting labs and other labs. And students will even receive a stipend for the summer to be part of this. So thanks to Emily and colleagues for this initiative. And this is also a new initiative that was proposed, a new paper by Raj Mori and Dan Stein. I don't know if Raj, you are on the line, if you'd like to take maybe a minute or two to explain what this is. If Raj, you're on, you're welcome to give a quick update on this.
Yeah, sure. Thanks, Paul. I mean, it's quite simple. We're really just highlighting the need for diversity in neuroimaging research. And, you know, being that ENIGMA is an international consortium, we thought that it would be a good place to start and get a sort of a survey of the representation of various diverse ancestries in neuroimaging research. And I should say that since, you know, this is not really based on genetic data, it's based on neuroimaging data. You know, we only know what the ancestry is from self-reported ancestry, and yeah, so and just to emphasize that you know, a lot of what we talk about as race, at least in the US and even some other countries, is really a social construct. So I just want to emphasize that. And we try to, you know, divide the categories in a way that might fit with on an international stage, because as you may know, the definitions can be quite different, like in the US versus Brazil versus South Africa, where race is defined quite differently. So you'll be getting a survey that Sophia will send out as a Google form, and you can just fill out the numbers on your cohort or your collection of cohorts. Thanks.
Thanks, Raj, and thanks so much to you and Dan for this great initiative. So just really to wrap up, I mean, there's only 5 minutes to go, just pointing to some emerging themes in ENIGMA. I mean, as you know, all these projects grow organically, and so these are just emerging themes that have been coming up. I think we're going to see a number of projects coming out on machine learning and deep learning and multi-site data. I want to mention a group led by Philip Seyman and Johanna Bayer and Lian Schmal, and until recently, Lee Jolens, looking at machine learning and harmonization. The interest group meets not particularly regularly, maybe every 3 months or so, to discuss concepts that come up in merging multimodal data, particularly looking at the site effect and adjusting for side effects. So anyone who's interested, please do contact Philip and Johanna. There's also an emerging use of resting state fMRI, which has been piloting, as you know, in the groups that we've mentioned. Very exciting potential to be sensitive to things that perhaps we were unable to see with structural and diffusion imaging. And also an extension, I feel bad for not mentioning them today, there's wonderful work by Alison Nugent on MEG and also Dirk Smit on EEG, looking at lifespan and disorder-specific changes in new functional data modalities for ENIGMA, MEG, and EEG. And also the efforts that we've mentioned to look into the cerebellum, work by Ian Harding and Rebecca Carestes with their tools for looking at the cerebellum, and also Matthew Campton's tools for looking at the rest of the brain using VBM. And just another thank you to the folks that contributed to the ENIGMA Toolbox. If you'd like to be involved in those papers, do reach out to Boris and Sarah. And that's another line of multimodal work that's very, very interesting indeed. A couple of final announcements, job postings. If you have interest in graduate student, postdoc, or faculty positions. A number of them are advertised here. If you'd like to post them, we'll collate and disseminate career opportunities both on the web and directly. So do email Sophia if you are interested. And I don't know if Sophia's on, if you wanted to just say a couple of words on the resources for chairs. I hate to put you on the spot, but you compiled all of these things. And I don't know if, Sophia, you have a moment just to go through some of the things that are available for all of the working groups.
Sure. Well, this is kind of our typical slide that we always include in the presentations, just as a reminder for the chair. So we have our Google Drive with all the chair's resources. So everyone should have access to that. But if you don't, for some reason, or you can't find it in your inbox, shoot me an email and I'll re-share the link with you. And this includes some of the, there's this overview document that is sort of like a README for the whole drive. So it links to other links and other files that are in the Google Drive. So it's just kind of to help everyone navigate through that folder. And then we have our typical authorship guidelines, secondary proposal forms if needed, authorship sign-up forms, examples. And also there's a link to the MOU that we shared back in April, and we also discussed this update during our June teleconference. So here's just a quick link to that. And if you need any help at any point setting up any of these forms or finding forms or anything else that we can help out with, feel free to send me a quick email.
Okay, thanks so much, Sophia. And there are also some resources here. If you have colleagues who are not members of ENIGMA, they're welcome to join in or inquire. There's newsletters and any number of frequently asked questions for people that might be interested in joining as to what sorts of things might be available to them. So again, thanks to Sophia for putting this together. So just on that note, I just want to say a huge thank you. I mean, we here at ENIGMA Support Group in Los Angeles, we owe a huge debt of gratitude to all of you for your leadership in not just the last year, but the first decade of ENIGMA. Let's hope that we'll have another very exciting year and decade to come. I want to say particularly thanks to the local team here in Los Angeles, Sophia, Netta, Chris, Lauren. I mean, there's a lot of people who work to support the ENIGMA projects here in Los Angeles, particularly Sophia, who many of you know has probably been on over 100 teleconferences in the last year and provided a great deal of support and coordination for these. So many, many thanks to her for all her support throughout this year. But again, just to conclude, we wish you a wonderful break. Hope you have a well-earned rest. And it's been a real pleasure and an honor to be part of the projects that you all are leading. And I'm sorry I couldn't quite summarize everything that everyone did in the last year, but hopefully it gives a little bit of a digest of some of the highlights for all of us. So I think that's it. We have a few minutes if anyone has any final remarks or questions, any comments. The floor is open.
Hi, Paul. This is Sarah.
Hi, Sarah.
So I'm just throwing this out there because Raj has inspired me, and it's something that I'm doing with a bunch of other consortium. In addition to being really interested in the diversity amongst our research participants, I think we should also be very interested in the inclusiveness and diversity and access within ENIGMA research community as well. So one of the things I'd like to get underway early in the new year as well is a questionnaire that might be coming out that looks quite similar to Raj's, but focusing on the members of your working group and who you are as a researcher as well. ENIGMA has grown in a very organic way, which means we often don't have set policies around certain things, but it is probably time that we take a step back and have a look at ourselves as a group and make sure that people are getting opportunities that need to be getting opportunities, and that things are, as best as we can, equitable and fair. So I'm just throwing this at Paul for the very first time right now, but I feel like there was some head nodding. So that will be a concept that will be fleshed out, and we will be in contact with you about this.
That sounds terrific, Sarah, and thanks again for your leadership over the many years with all of the very difficult projects that you've brought to fruition. There's time for some final remarks. We could also, I was so excited that so many people here, we could also do a photo. Now, I realize it's not very convenient for anyone who's in bed, but if anyone would like to put the camera on and be featured in a photograph, if you are in bed, as I sometimes am at 6 a.m. when your calls are going on, we'll substitute a recent Zoom photo and that would probably be fine. I think, obviously we wish everyone well in coping with the whatever's going to happen with the pandemic. But one way or another, I think we'll meet obviously virtually as well.
Oh, quick question. Can you hear me? This is
yes,
David.
We can. Hi, David.
Happy holidays to everyone. I actually was doing a talk about the time difference. I was giving an international talk last night at 1 a.m., ended at 2:30 a.m. But one of the questions that came up, and I was so happy we had the call today, I was bragging about ENIGMA. It has to do with sports and concussion. But someone asked me, is anyone in ENIGMA looking at neuroimaging as a biomarker predictor for long haulers in COVID? It had nothing to do with what I was talking about, which was sports concussion. But, and I said, there are many, many ENIGMA groups. I didn't have the answer. I did refer them to Sophia, but they were wondering now with, kind of the, death of, of neurons and all the things that's coming out in the late press. Is there any neuroimaging that will help us predict people who might be at a greater clinical risk group for long haul symptoms, whether they're neurocognitive, so I said, I'll ask the Grand Poobah tomorrow.
David, I don't know if Emily or I don't know if Carrie Sopenko is on. I mean, there's an ENIGMA COVID effort. Maybe Emily, you have a very good perspective on this. And I'm not sure if Maheen is on, but Emily, you have a good perspective on this.
Yeah, if Maheen or Carrie are on, they would have, I'm trying to see if they're on here, but.
Hey, I'm here.
Wait 1 minute.
Oh, hi, Carrie.
Hi. Yeah, David, we've got a COVID-19 working group. It's been a little bit riding like the pandemic for how much we've been able to meet, I think. But we have a meeting coming up right after the holidays. So if anybody's interested in it, we can add you to the list. It's still a little bit slow going at the moment, but we're hoping to really hit it off in the new year and get moving on that.
Yeah, the reason I got involved is a number of the athletes. I'm going to be going over to Beijing with the U.S. team, and with all the sports, and I take care of a number of pro leagues. And now with all the canceling games, I'm starting to get questions literally from athletes saying, should I be looking at these effects long term? And I don't really have the answers. The other thing that's been interesting related to this, and I need to get the reference on the new group looking at some advanced transcranial magnetic stim. The Stanford group's got some interesting data out on it. It's much better for short-term depression. The athletes are all over this. So they've asked me, and I said, I've got to go to the real experts. My only expertise is I get the joy of being able to work with the people who are doing the phenomenal work. So a lot of interest in the sports world because a lot of these athletes are testing positive now. And just like with concussion, they're worried, is this going to be a post-concussive like, and I said, I'm sure we're studying it, but not me, but I'll try to find out who is, and I'll try to be that liaison.
Yeah, for sure. Send me an email. I think from the neurological standpoint, we're all a little bit confused on what it's going to do. And I think, there's now questions about whether or not vaccinations change it and whether the different variants have different effects on it, since they seem to have different severity versus, how quickly they're spreading. But I honestly don't know that we're going to know that for very long. We just published with the Big Ten COVID registry a paper in JAMA on myocarditis. So like those have a little bit more understanding on what's happening, but from the neurological perspective, I think.
Well, that's great. I don't want to lengthen the meeting, but I'll reach out to you so I can reach out to them. I was amazed at the level of interest in the sports world about this because the COVID thing has become, and that's going to be the big thing in Beijing. I mean, I'm dealing with them. So I'll reach out to you, Carrie. Thanks so much.
Okay.
Sorry to lengthen the meeting.
Thanks, David. And thanks, Carrie, for your leadership, and also Maheen, too, for all of the work that you've done in that area. So we have 1 minute to go. I don't know if there's any final remarks or questions or announcements that anyone wants to make. All right, it's wonderful to see everyone. Thank you for taking time in what's a very busy week, and hopefully you have some rest and relaxation with friends and loved ones. So we'll see you next year. Thanks so much.
Happy holidays, everybody.
Bye.
Happy holidays. Bye.
Bye.
Happy holidays.
Thank you.